webinar series with Dr. Elaine Ingham. If you're watching this on YouTube, please feel free to subscribe. If you're on Facebook, give us a like and share this content. Once again, we are Economic Action Team. So if you're watching this, if you're watching this on Facebook or YouTube, I recommend that you hit that like button right now and subscribe to our channel so that you get videos like this every time we go live. So today it's going it's going to be Dr. Elaine Ingham, the Soil Food Web, and yeah, this is another Q&A session. So those of you who are live in the audience, and those of you in the YouTube channel watching this or watching this over Facebook channels, um, welcome to all of you, and hope you're all doing well. So hit that like button, hit that subscribe button, and that'll be awesome. So you'll get notifications every time we come on. So those of you who are here live, thanks for joining us. And those of you who are watching this on YouTube or Facebook, so this is being telecast, or not telecasted, but this is being um, simulcasted. So simulcasted over several platforms, and you might be watching this live right now on Zoom, or you might be watching this on YouTube or Facebook. So go ahead and hit that like button, subscribe our channel, show us your support. So you want to see every time we come on live with any of our amazing speakers, like we're going to do today with Dr. Elaine. So in a little bit, we'll start this webinar. This is a replay. Many of you have seen this before, but if you have seen this before, it is I'm sure you'll enjoy the second time. The second time, if you have missed something the first time you watched this, take notes, visit her website, get more knowledge. And if you haven't seen this, this will be amazing. So without further ado, let's get started with Dr. Elaine Ingham, her Soil Food Web replay. And also don't forget to put in your comment, type in where you're at in the world, and if everything is coming in, the audio, the video, and everything. So let us know. All right, so here we go.
Hey there, everyone. Welcome to the session. I know my bad, there was a little bit of a mix-up there on the email versus now. I said 11 on one of the emails. And so for folks of you who have been waiting 30 minutes, thank you for your patience. I know maybe you were throwing your food at the wall like, why did this, he duped me. He duped me, and now I'm been sitting around for 30 minutes. Thank you for your time. Well, this is going to be great. We've had Elaine joining us again for a fantastic session where she's going to answer your burning questions about soil biology, compost, the classes, bioremediation, all these great things which you emailed before and answered about. Just in case, we've included some of the questions we're going to answer on the handouts tab, which you can look at below. There's also, for folks interested, that big, awesome, the half-off discount ends tonight, and the link for the classes is also in the chat box. So the way this is going to work is, we're going off all the folks who answered questions before. If we have time, we'll allow people to ask more questions. However, we got a lot of questions to go through, and we're going to answer them if you're here. If you're here, awesome. We'll get to the question you submitted. If not, we'll have to move to the next question. So without further ado, let's get started. Thanks, Elaine.
Yeah. Glad to be here. So got my question answering skills all honed this morning.
All right. Oh, we got Neil here too. Hey, Neil.
What's up?
Good morning, morning.
Are we recording already?
Yeah, it looks like, let me give a little check. Yeah, we're recording, so we are all good. So,
All right.
So how about this? I can read. Elaine, would you prefer just going with questions, or I could read them if you want?
Yeah, that gives me a second to think about what I'm going to answer.
All right, fair enough. Okay, this first one's from Leonard Barron. This is the general questions. He's saying, can nutrient-dense foods be grown with the soil food web and liquid solutions using vertical farming systems?
And the answer to that is absolutely. That's totally possible. When we're dealing with liquid solutions, so like nutrient troughs or float tables or flood tables, or where the nutrients are being added in the liquid. What are the kinds of nutrients? So let me quick go through the general concept with these vertical farming systems or tables or hydroponics, aquaponics, something like that. You first of all want to get the biology established on the root systems. And so typically what we're going to do is put into your liquid source a good compostee. So now all the proper organisms are going to be moving past the root systems of your plants, whether that's coming vertically or whether it's the float table or it's coming through nutrient trough. The biology is moving by those root systems, and of course the bacteria and fungi are going to glue themselves to those surfaces, start making aggregate structures, which then allow the protozoa and the nematodes to find habitat along that root system. As soon as the protozoa and nematodes start doing their jobs of consuming the bacteria and fungi and releasing the nutrients right there at the surface of the plant's going to start getting all the nutrients it requires. So then you need to be adding a liquid food resource for the bacteria and fungi that contains all of the nutrients that your plant is going to need. And it's always, it's kind of back to that question of, well, how many nutrients does your plant needs, and in what concentrations? Well, what's going to balance the soluble forms of these nutrients? That is what the plant takes up. And so that's the biology around that root system. The plant puts out the exudates, feeds those bacteria and fungi that go forth and look for solid materials, organic materials, that they can pull the soluble materials they can pull straight out of the liquid running by. So what is the food resource that's going to contain all of the nutrients that the bacteria and fungi and then ultimately your plant need? And that's a humic acid. So yes, I know we're in the middle of a controversy in the soils world about humic acid does not actually exist. Well, when you're using strong acids and strong bases to extract something from the soil, that extracted form doesn't actually exist in the soil. What exists in the soil is that material before it's extracted by the strong acids and strong bases. So if we go ahead and call that material humic acid or fulvic acid or ulmic acid, those are the things that comprise humic acid, or should we say humus? Well, except humus contains a lot of other stuff as well. But we're extracting that material, or you can take from your compost, you can just passively run water through compost, and you're going to extract the water-soluble complex food resources called humic acid, for lack of a better word to call that material, or liquid water-soluble humus is what we're extracting. So you put that water-soluble humus into your tank, in your vertical flow system or your hydroponic system, whatever. And that's what the bacterium, fungi, their enzymes happily waiting for that humus to go by to grab the nutrients and pull those nutrients into their bodies. And then the bacterium, fungi are eaten by protozoa and nematodes. And that releases nutrients in that plant available form right there next to the root system in all the proper balances. So yes, nutrient dense foods can definitely be grown in liquid solutions using vertical farming systems.
Okay, awesome question. All right, the next one is coming in. Let's see if she's here. Let's see if Doris Bancroft is here. Oh, she is. Okay, great. So she's submitted this question. She says, hi, just wondering what effects, this is good, well, what effects prescribed burning of our forests has on soil ecology? These fires get pretty hot and it take a while before the grasses come back. Are we possibly doing more harm than good? Great question. That's really good, especially with all the fires happening. Oof.
Yeah, when they're careful about the time of the year, when they time those fires correctly, there isn't a lot of damage done in a controlled burn or in a prescribed burn. Typically, they want to allow the fire to burn very rapidly across the surface of the soil, lightly burning that mulch layer, the organic layer that's developing, burning some of the understory, shrubby grasses, weeds, things like that. And as long as that fire is pretty rapid, pretty quick across the surface of the soil, so that the temperature of the soil does not increase markedly with depth. So as long as a very fast fire, very rapidly moving across that surface, it doesn't stay in flame for that long, your soil system can tolerate, temperatures up here where the plant material is burning, quite high temperatures up here. But as long as the temperature of the soil doesn't get very hot for very long, it's when we have charcoal, when we have like a log, or large concentrations of debris on the surface of the soil or even into the soil, when that burns and the coals from those burning materials sit in the soil for a long period of time. So think of the fires in California this summer, where you could still see smoke coming from the soil. You could, at night, you could look across that hillside and you could see areas of high concentrations of smoldering coals still burning for a long time. That's where the real damage is done. I think back to the work that was done in Yellowstone National Park, where they were looking at the effects of the amount of heat and for how long those temperatures had continued on in the soil. The longer that's hot, the higher that temperature is, those two factors, that's what's going to determine whether the soil is going to be basically sterilized and become dirt. There were places in the Yellowstone fire and in many fires, really hot fires, with heavy fuel load on the ground. The silica in the soil will melt and the soil surface becomes glass. And those are horribly damaged soils. Those are massive problems. Imagine rain hitting a surface like that. That rain just runs right off, and now you've got the start of a mudslide. Hillside failures, slope failures are going to happen. So for a very short period of time, hot fire, not a problem. It's where the temperature at depth into the soil gets hot. That's where you're essentially sterilizing your soil. And now where is that source of inoculum going to come back from? Prescribed fires are rarely very extensive. They're only doing maybe 10 acres, 20 acres. Think about how long it's going to take microorganisms from outside that 10 or 20 acres to move into all of that 10 or 20 acres that were burned. It's probably only going to take 6 to 9 months for all of that to be getting colonized. Think about like the Thompson fire in California, where they're looking at 20-mile width areas that were burned. How long is it going to take for biology to get back into the middle of that 20-mile area? So that's the return of those organisms. Should we be sending helicopters or airplanes out with really good compost tea to reestablish the organisms in those areas? And that's exactly what we should have done in California or Oregon or Washington or Utah, Nevada, all of New Mexico, where there were bad fires over extensive areas this summer. We should have reestablished this biology in the soil, so that biology would be now holding the soil in place, keeping the runoff, stopping those mudslides from happening. But nobody thinks about that. We need to rev things up one more. So hopefully that answered that question.
All right, that was a great question. I'll probably buy like 100 boxes of smoked salmon, or for the first person who's the compost tea firefighter, because we need that person to step up. That'd be awesome.
Yep.
Okay, so the next question is from Kate Radloff. Kate asks about hops. This is we're on to the plant questions now. She says, have you ever worked with hops before? How would you recommend a conventional hop farmer to make the change from chemical to biological farming? Is it possible to make a slow transition in herbicides, or is it all or nothing process? And her second add-on is the right fungal bacteria ratios for hops.
Okay. The correct fungal to bacterial ratios for hops are up in the shrub category. Hops are really vines. They can get fairly woody, so that those thick stems that have a very definitely woody characteristic to it. And so what we see then around the root systems are fungal to bacterial biomass ratios around 2 times more fungi than bacteria, up to 5 times more fungi than bacteria. And really, when you start looking at that, normally what we see, hops are going to have somewhere around 800 to 900 micrograms of bacterial biomass. And then you need to double that or quadruple that for the fungal component. So, you want around 5,000 micrograms of fungal biomass for 1,000 micrograms of bacteria. You need to have somewhere around 50 to 10,000 to 50,000 protozoa, and of course you want the flagellus amoebae. And then you want to see the bacterial feeding, fungal feeding, and predatory nematodes. You want somewhere around 100 to 500 nematodes per gram of your soil. You want no root feeding nematodes, of course. There's a good indicator when your hops aren't healthy that you'll have a lot of root feeding nematodes. So let's get that switched out. And then of course with hops you want microarthropods as well. So you want some of the cute little Collembola, the mesostigmatid and the prostigmatid mites, because they do a really good job of taxicabbing these really beneficial organisms around the root system of your plant. So have we worked with hops before? Yeah, with large hop operations in a number of places around the world. How would you want to do the change? First step is, of course, make good compost. So we have to do the training with people who want to do the conversion so they understand how to make good compost, and they always have visions of, I've got to make 10 pounds of compost per acre and spread that out there, and oh, I can't make 200,000 tons of compost. You know, they do all the little calculations, and so I was like, no, 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 no. What you want to do is make small piles, the half a ton, quarter ton piles of compost, and make sure that you make really good compost where you have really good inoculum of bacteria, fungi, protozoa, and nematodes. And then you inoculate that into windrows where you put those organisms into essentially a sterilized organic matter. And you bring along that really beneficial set of microorganisms, and then you will typically make a compost extract or a compost tea for actual application out onto the soil. So in the fall, putting out an application of compost tea or your mulch that's been enhanced by getting the right biology, or if you can, put out solid compost. Typically a ton per acre for the compost, or you want to be, with the teas or extracts, you want to be replenishing whatever's missing in your soil. So you're looking at, you know, maybe somewhere around a couple gallons per acre. It's an inoculum going out. Then all through the dormant season, you let that grow and do its job of building cell structure, starting the nutrient cycling going, get your understory plants established in that fall period. Next spring, you check your biology, add back whatever's still missing. And with hops, as it starts to regrow, at bud swell, you would want to apply an application of the compost tea so that you're getting those organisms stuck to the surface. A month later and a month after that is typically the transition. Now, if you have growers who are like, oh, I just don't know that you really can do this. I don't believe that the organisms are going to be able to cycle enough nutrients. What you say to them is, okay, reduce your inorganic fertilizer applications by half. You know, so reduce it by half the amount. Well, okay, and if they see that it's not quite enough nutrition, we'll go ahead and we'll put on a fish hydrolysate. We'll put on something that contains a high amount of plant available nitrogen so that they won't have any fertility problems. I've actually never had to do that, because if you get the biology into the soil, nutrient cycling is now going to be cycling every nutrient that the plant needs, not just nitrogen, not just phosphorus, not just, you know, the few things we tend to put on as inorganic fertilizers. So we're going to put on the whole shebang. So I've never had to go back in and apply like a fish hydrolysate or, you know, a fertilizer application. But you tell them we can do that if it's needed. Sometimes we have to go back in and do the compost teas more than 2 or 3 times because they have an overwhelming disease problem. So if things get really bad, okay, we'll let them apply fungicide or an insecticide, but then look at what that disease-causing organism is or that insect problem is, and do all of the work necessary to wipe out the eggs where they get laid in the fall, where the eggs hatch and where are the larvae, and make certain that you reduce the number of larvae that make it into adult mode, and then make certain that you're putting on the biology that's going to kill the adults long before they can get around to laying eggs, so that you will never have that insect or that disease-causing problem ever again. So it is possible to do the transition a little bit slower and still allow use of fertilizers and herbicides. Typically, you want to wait until the weeds start showing themselves. Because, you know, often we've had the problem with people who, well, we just go out and we put out the glyphosate, you know, 2 weeks in advance of germination of the weed problem, because we're never quite sure what the weather is. and we don't want to have any weeds. So they're unnecessarily putting out herbicides. They don't even know that the weeds are going to germinate and grow, and they go ahead and treat for them. Well, make them wait. Let's wait until we actually have observers out there showing that there is a problem developing. So in that first growing season, more of the emphasis might be on scouting problems. Recognizing that that's starting to happen, and yes, we're going to have to go ahead and use an herbicide or an insecticide or a fungicide to deal with the problem. But in the second year, again, you put your money into doing the scouting. We rarely, if ever, have to be dealing with an herbicide or any insecticides, pesticides in that second year if, and please listen to this, if you've gotten the biology correct in the soil. If you haven't gotten the fungi up to where they need to be, then yes, you are going to have to use an inorganic fertilizer. Hopefully way, 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 way less, but you may have to use something like that if you don't get the biology right. Okay?
All right, thanks, Kate. I know, Kate, we helped her build a really awesome natural swimming pool out in Iowa. So appreciate her writing in. So next question. All right, now we're on to the bioremediation questions. These are actually most of the people asked about bioremediation as a focus, like, how to break down diseases, how to deal with toxic chemicals. This is kind of the bulk of the questions. So we might spend a bit more time on here than most. So the first question comes from Katie Van Gorder. And we'll see if she's here right now. Okay, she is here. So she says, I was wondering if there's a chelation trite treatment, something like a phytoremediation plant, planting certain types of plants that can uptake specific chemicals, and by doing so remove toxins. So she's wondering if there's a specific phytoremediation plant to remove excess aluminum sulfate from topsoils.
Aluminum sulfate is typically an anaerobic condition problem. So that's really what we want to work on at the same time as perhaps absorbing or taking up some of the excess aluminum sulfate. Well, where did that excess aluminum sulfate come from? Well, it came from exchange sites on the surfaces of the sand, silt, clays, rocks, and pebbles. It's there in the crystalline structure of the mineral component within your soil. We want to put it back there. That's where it came from. I want to put it back there. Let's put it back in storage mode. And so that means we have to get rid of the anaerobic conditions. We have to change the pH so that the aluminum is now going to adsorb back onto those surfaces, and what's going to be released is the calcium, which is what we want. So how do you remove the anaerobic conditions? You've got to build structure in the soil. Now, probably the fastest way to get started on that is to, yeah, I know it sounds pretty bad, go till for the last time ever. So if you're in an annual system, that's easy enough to do. Let's put on a nice, maybe an inch of compost on the surface of your soil, and then till that in. Please make certain that you're tilling it in below the compaction layers that are probably in the top 4 feet of your soil, or 1.5 meters within your soil. So till the biology into the soil. So now you've got something in that soil that will start to use the organic matter and build structure. So oxygen can be moving all the way through that profile. At the layer that you left still compacted, and actually probably increased the compaction a little bit by the tillage process. The tillage equipment presses down on the soil, and so that is compacting at whatever depth that tillage equipment, your discs or your chisels or your bars went down. You're compacting at that depth. But it hasn't been compacted for long, we hope. So the microorganisms, where we put that compost with all that really good set of microorganisms, they will immediately start chewing up wherever that compaction layer was imposed, and they start rebuilding that structure. So long before your roots get down there, long before water infiltrates all the way through, that structure will be reopened, And so normal infiltration of the water can happen. Storage of water through the whole depth will happen. So as you get oxygen then moving down through, your aluminum is going to be reabsorbed into the crystalline structure of the rocks, the pebbles, the sands, the silts, the clays. So that aluminum sulfate is now bound in the proper place because the pH is now up above 5.5. We're back in aerobic conditions. The aerobic organisms do not produce the toxic acids that were causing that problem with the aluminum sulfate. So one of the other things you can do for an immediate improvement, if you've got these kinds of anaerobic waste compounds causing you problems, is to put organic matter into the soil. Organic matter, hopefully, that has a lot of surface area. And one of the best organic acids to be adding is, of course, humic acid. It's one of the most complex organic materials. It's soluble, so just get the, use water in a good compost to extract your water-soluble humus and inject that or put that into the soil, and that should be able to absorb any excess aluminum sulfate that is present. Just remember that you can't expect the aluminum sulfate to remain absorbed to that humus material if you don't add biology, because it is the microorganisms, the bacteria and the fungi will take those toxic materials and put them back into the structure of the organic matter. Get them tied up where the half-life on those materials is around 500 years before they will be re-released. If you don't have the biology working on these things and putting that absorbed material on the humic acid back into the physical carbon-carbon-carbon bonds, getting attached and put back into that structure, then the half-life on that absorption is only about 2 weeks, and you start seeing the problem come back. So we've got to get the permanently operating beneficial aerobic organisms back into the system to deal with those problems. So there are certain plant species as well, so you've got a lot of choices in which approach are you going to be taking here. You can choose to use different kinds of plants, but it's not really which species of plant is the best at taking up these things, because we don't really want the plant itself taking up those toxic nutrients like way too much aluminum or cadmium or any one of the lead, so all those problems. We don't want the plant to be taking that up and putting it into the fruit, putting it into the seed, putting it into its body, because then we have consequences of using that material if it's high in those toxic, those heavy metals, the contaminants of different kinds. So when we're talking about heavy metals here, what you really want to find are the mycorrhizal fungi that will take up and sequester those heavy metals in the fungal biomass. And typically what happens is, you've got the root system here, mycorrhizal fungus moves in and is being fed fungal foods from the plant. The direct connection with the exudates, instead of the exudates being released into the soil, they're directly fed to the mycorrhizal fungus. And the plant says to the mycorrhizal fungus, look, I'll give you this energy here, I'll give you this food, if you promise to go out there and make sure no lead makes it to the root system. So the plant doesn't take up the lead. The mycorrhizal fungus takes the lead up and stores it in the mycorrhizal fungi that's just outside the root system. It's basically like the plant says, oh, no, I told you I don't want any of that stuff. You keep it out there. And the fungus goes, okay, and stores that in the hyphae outside the root system. So when you take this plant that is accumulating It's really the fungus that's doing it, but well, okay, it's a plant-fungus interaction, kind of part of the same body. So when you take that plant out, you want to get as much of the root system and those mycorrhizal fungi as possible. So you want to be gentle about this. Now, you don't want to take the stem and rip the plant out of the soil, because most of those mycorrhizal connections then are left in the soil. You want to dig underneath the plant and gently lift the plant out of the soil, shake the soil gently off the root system, and then you've got all of that metal, heavy metal, contaminant problem sequestered, and that mycorrhizal fungus. You can take that plant material, normally, because none of the excess lead or problem contaminant is actually moved into the above ground part of the plant. You can cut the stem off at the soil surface. You can go do whatever you want with that plant, that part of the plant. It's that root system that you would take to an oven and burn off all of the carbon material, leaving you with pretty much a pure metal, whatever it was that was the contaminant. You now have collected a product that you can sell to other people. So hopefully that gives you an idea of some of the different ways to approach this problem.
There, that's awesome. Sarah Ozawa had also asked about that, how you tie up toxins like lead in urban industrial areas. But I think you answered her question as well as answering that question about aluminum sulfate. Anyway, so yeah, Sarah, hopefully that answered the question. Let me know if that did your question justice about cleaning up lead toxins. So the next one was from, this is about allelopathic compounds, how to break them down in your compost. This is from Anne Malloy. Let's see if she's here. She says, yeah, all right. Anne says, how do you effectively compost trees that have supposedly poisonous compounds in them, i.e. pines, juniper with rosins, eucalyptus?
What you're going to do is typically take that woody material, you're going to chip it up so that there's a greater surface area exposed. You want to then let that wood chip material be in a big pile, or spread it out on a tarp or something, and you wait until that strong smell of the pine or the eucalyptus or the juniper, you lose those volatile compounds. So all those resins that like juniper may have eucalyptus, pines, black oak, black walnut, all of these things, really strong smells. Even oak has some of these terpenes and tannins. That strong smell, that's the antimicrobial agent. You're smelling those compounds as they're being volatilized and released into the air. So you want to make sure that all of this plant material gets aerated. And once that strong smell is gone, then it's just like any other plant. The antimicrobial agent has been dispersed. And so now if it's, it's not smelling very strong, you can have a little bit of pine smell, but it's like when you pick up that chip of, the branch of the pine tree and you're smelling, okay, I can't smell it, oh, I'm getting a little hint of that pine smell, fine. Most of that antimicrobial material, the poisonous compounds are gone. And so it's not going to cause a problem in the compost pile or whatever you might want to do with that woody material. The antimicrobial agents are gone. So it's just having the patience to let the antimicrobial agent volatilize off. And of course, the smaller you chip things up, the faster the release of that material from the wood itself is going to happen, because more surface area, more area for that volatile compound to be degassed and lost from your wood. Okay?
All right, fantastic. Next question comes from Eric Knight. Eric's asking, she mentioned about the failed herbicide Imprellis. He says he focused on the debate very closely. He says biochar was used as a remediation agent, but that didn't really work. But he says, how many attempts were made to remediate the effects on mostly golf courses when this failed herbicide killed trees en masse and cost DuPont $500 million plus in fines and settlements? Did Elaine actually do trials with compost biology to remediate the tree killing effect?
Nope, there was absolutely no interest on the part of either the USDA or DuPont to utilize a microbial approach. Or if they did try, I was never aware of any of that work. So as far as I understand, they used biochar to try to have the microorganisms utilize the Imprellis, chew it up. Well, it's not just microbiology in general that will deal with the Imprellis. And it's only probably about 1,000 species of bacteria that are actually capable of breaking down this synthetic herbicide. Well, it's synthetic. Most organisms have never seen this set of bonds in just this particular fashion. So it's going to be a limited set of microorganisms that are capable of using it. Only 1,000 species. So it's actually a fairly good diversity that you can have. In most compost piles, we will have some of these species growing. They're usually from the group that we used to classify as Pseudomonas. So I can't always remember all the other names that the Pseudomonads have been dispersed into, but there are other people on the planet who keep track of those things. So what they should have done would have been to take the charcoal from making that process of making biochar. You first have to take your organic material and subject it to very low oxygen levels at very high temperatures in order to make it charcoal. Well, in that process of heating and lack of oxygen, all of the microbiology, all organisms, anything living in that material is now dead. It's been put into a crystalline form, basically. If you just take biochar and put it into the soil, all you're going to do is suck up nutrients into the charcoal to try to balance the high concentration of carbon in that material. It's a crystalline structure. You have to have microorganisms in order to pull the nutrients that are in biochar into a plant usable form. So they didn't put the biology back into the biochar. They just made charcoal and put charcoal into the soil. And so of course it didn't work. Of course they couldn't decompose the Imprellis. Let me think. Decomposition. What do you have to have in order for decomposition to actually occur? It's not a magical process. It's not something where you just stick some organic matter and it gets converted into something else. You have to have the organisms that do that job of converting it into something else. So what should they have done? They should have actually used biochar. If you have no biology in that charcoal, then call it what it is. It's charcoal. It's not biochar. No biology, no bio. Can't call it biochar. So of course it didn't work as a remediation process. How many attempts have been made to remediate effects in golf courses where the herbicide has killed things? I'm not actually aware of any time, any experiment where they have done work to get the proper set of microorganisms and the foods to feed those microorganisms out where they would be capable of decomposing this synthetic herbicide and getting rid of the problem. Certainly with clopyralid, there were a number of studies that were successful. I think I probably have a few of those studies on my website under, remediation, bioremediation. So you might go check our websites. At least you would get the information for the clopyralid. I don't have that. I don't think I have any studies on the Imprellis because they never did any studies with biology. You get to a certain part of the USDA and they disregard anything having to do with biology. It's, biology doesn't really do anything. It's not really important. I've been in this work for 75 years and I've never heard anything about this, right? When they went through college, there wasn't anything about biology when they were at university. Biology wasn't discussed 50 or 40 or 30 years ago. Only if they've managed to keep current in the scientific literature over the last 30 some years would they know about this. And it just didn't penetrate when we talk about the Imprellis. Okay. So did I actually do trials with compost biology to remediate the tree killing effect? Yes, we have, but I've never published them. It's one of my lacks at this point in my life is lots of data, not enough time to publish. So see, that's why I'm expecting everybody else to do that. Okay.
That was awesome. It really goes. There's a frightening amount of chemicals out there that people are just spraying on the land with no idea how to break it down. So I'm glad you guys are tuning in to learn about how we could actually do something to address this process, especially with things like glyphosate, which we know are getting into our systems and they're getting into our urine, they're affecting our health. And so we got to know how to protect ourselves and our land against them. So now we're moving on to the compaction questions. So these are questions more so about getting through compaction layers, things of that nature. So the first question, let's see if Liam is here. First question is from Lem Huang. Lem asks, how do you punch holes in the ground? My compaction level is between 1.5 to 2 feet. Below it is really hard compacted clay. He tried using a steel pipe. Under water pressure didn't work. You can only go down 1.5 feet. He says the clay under that is too hard.
This sounds like you're really going to have to go in there with a deep ripper of some kind, a keyline plow or yeoman's plow. You're going to have to use some big mechanical equipment because this sounds like it's really nasty. You've had compaction happening for so many years. There's probably a lot of chemical bridging that's happening so that this material is just like concrete down there at 1.5 to 2 feet. So you're going to have to invoke some really big equipment. One possibility to avoid having to use the big equipment would be getting injection system where you can push that metal rod down into the soil as deep as you possibly can go. And maybe it's only going to be 1.5 feet, or sometimes it'll be 2 feet. Sometimes you're going to find a crack and you're going to be able to push that a little bit way down into that really hard compacted hard pan. And then at that point, you open up the flow of the compost extract into the soil, and you just let that compost extract pour out of your injection equipment. I'll give it 10 seconds, 15 seconds, so you are putting a lot of biology into that area down in the soil. Hopefully that biology will be able to slowly get absorbed by the surrounding material, and all that, those organisms will be able to start building structure for you. As you pull that injection equipment, the tip of it, as you pull it back up out of the soil, keep pouring real good biology into your soil so that whole 1.5 to 2 foot depth of your soil is going to get inoculated with microorganisms. If you have good structure in the soil, you probably only have to go every 5 feet for injection the first time through that you do this, because from that injection point, This biology will be able to move out approximately 2.5 feet in every direction. So then the next point that you inject is going to be 5 feet away. Then if you ever have to repeat that ever again, then you can go further and further distances. But usually a one-time application where you're doing that close injection only has to be done initially. As long as you do nothing to kill the biology going into your soil, then you should never have to repeat that. But it sounds like there's probably 70 years worth of compaction in this soil, and you are gonna have to take a little bit of time to remediate it. If you can get in there with deep rippers or subsoilers, keyline plow, yeoman's plow, to break up that compaction layer so you're getting your biology down to 2 feet or 3 feet or 4 feet. 4 feet is about as deep as I've ever seen a keyline plow go. But if you can do that, then and get the biology back into the soil. So typically what we do when we're doing tillage, if you find that tillage equipment, is we'll put our compost on, and we tend to put it pretty thick on the surface of the soil, like 1/2 inch, 1 inch, and then you till that in, and you want to till down as deep as you possibly can go, and that's as deep as you possibly can go is probably about 4 feet. Okay.
All right. Great question. Thanks, Lem. Moving on. We're gonna move on now to the well, it's more. There's one more compaction question. Hopefully we can get through this kind of quickly. It's about the fragipan layer. Abigail asks, can soil biology help break up natural hard pan, specifically a fragipan layer?
Sure, it's no problem. It's going to be exactly similar to what the previous question was. I don't ever, natural or unnatural, human imposed. I find most compaction is actually human imposed. So you have to go back in time and find out what somebody did 100 years ago, 150 years ago to cause this layer. Sometimes in soils that are moved by water, you'll get a layer of clay or a layer underneath a stream or in a lake or a pond or something. And that's just breaking up that compaction layer, getting the good biology in there to rebuild structure so that the water will drain, so you don't have that puddling. Now, most of the time when you're dealing with compaction of any kind, but compaction caused by whatever it got caused by, water moving through the soil will infiltrate downward until it gets to that compaction layer. And then the water starts backing up because it can't go anywhere. It can't infiltrate any further. So you get a puddle developing. You've got a big mud pie starting to happen. If there's any slope, then the water will drain off in one direction, but unfortunately it's going to take a lot of your soil and all of your soluble nutrients with it as it goes. So it's critical to get that broken up, physically broken up. If you don't have the tillage equipment, and this is, remember, one last time that I'm recommending tillage, you should never again have to use this tillage equipment. So don't buy the tillage equipment, rent it. Or if you can't do the tillage, then the injection, and you may have to put those points where you're pushing the injection tool down into the soil. You may have to go closer than 5 feet, maybe 2 foot between injection sites if you really want to break up these kinds of fragipans. Okay?
All right, thanks, Elaine. Next question is from Julie Withers. This is about dealing with horse manure. Sorry, dog. There's a dog trying to get in here, but can't get in. Okay, so Julie Withers is saying, I'm tending to a number of fields grazed by horses. I'm aware that a number of horse diseases are on the rise due to the decreasing nutritional value of grass. So she attributes these diseases that are coming in horses to poor nutrient cycling and poor grasses. So she's wondering about the best way to compost horse manure on site so that it can be returned to land brimming with microbial life, more particular fungi, which are most notably absent in the soil. Any ideas?
You're going to want to make that compost correctly. So typically what we do is start with a basic recipe, and then you will adjust it for the actual based on the temperatures that actually occur as you're composting this material. In order to deal with the diseases and the pests and the toxic, you name it, the weed seeds, all the undesirable things in a compost pile, you've got to make sure that all of it's getting processed by earthworms, or you've got to get the temperature high enough to kill all of those things that you don't want to have in your pile, but not kill any of the really beneficial organisms that will survive the heating process. So typically 10% of the pile in the summertime, when temperatures are already pretty warm, only 10% high nitrogen in the pile. In the winter, we up it to 20% high nitrogen, and that's your horse manure right there. It would be preferable if you were not treating your horses with any kinds of deworming medicine. So that deworming medicine is a general sterilant, and it kills off most of our good biology in your horse's digestive system, and so the manure coming through doesn't have any organisms in it. That means the rest of your starting materials for the pile has to have really good levels of microorganisms in it. So you take your horse manure, 10% of your pile is going to be horse manure. You want to mix 30% of the pile should be a mix of plant material that was cut when it was green. After you cut it, it's fine to let it dry down, and it looks kind of brown and not so wonderful. It doesn't look green anymore, but you haven't lost any of the sugars or the proteins or the carbs as long as you've kept it good and dry. Microorganisms require water, just like human beings. So keep it nice and dry, and you'll keep all your good nutrients in that plant material. And then the rest of the pile, about 50 to 60% of that pile, should be woody material. So stalks of corn or woody stalks of different kinds of materials, wood chips of different kinds, chip them up, fairly small. Brown leaves that fell to the surface of the soil in the fall, residues of any plant that's actually gone brown while it was still connected to the root system. That means all of the nutrients that were in that plant got translocated into the root system, so the above ground standing dead is woody material. So then you want to follow the temperature that evolves in that pile. Temperature is a result of the microorganisms growing. So as they reproduce, heat is produced, just like human beings. A lot of temperature involved. You want the temperature to get above 131 degrees Fahrenheit for a full 3 days before you turn that hot middle of the pile and spread it on the outside. Then what was on the outside of the pile, you want to get it into the middle. So it usually takes 2 to 3 turns to make certain that everything has been in that hot middle of the pile and you've got it hot enough, long enough, so that you've killed all the problem organisms. So then once that pile cools off, and if you've done this right, it only takes about 21 days, maybe 30 days for this pile to get back to ambient conditions. If it's taking longer, your pile went anaerobic for a period of time and you grew some not so wonderful organisms in that pile. So you can see that for about 10 days, it's really going to take constant attention. Every day you have to be paying attention to the pile and making certain everything's going just fine. So put that compost out, spread it at about 1 ton per acre when you're dealing with pasture. Typically that's all you need is just that inoculum. Put the compost on either in the fall or in the springtime. The rest of the year, like 3 times then through that spring growing season, you take whatever compost you have left, extract that biology into water, and then spray that out in that liquid form, which is way easier than spreading the solid. And that way you get the biology back into your soil that will do the nutrient cycling. And your plants will finally start getting all the nutrients they require, which means they're not subject to diseases or pests. The weeds do not do well at all. Let your weeds be the sick, unhappy, unhealthy things. Yes. Let the plants you want, the really highly productive grasses. And then what you see is that actually bricks will go up. But when you're looking at bricks, it's not just sugar that you're measuring when you're looking at bricks. It is the sugar and all of those solids that can be in the sap material. And so you're looking at proteins, you're looking at amino acids, you're looking at a number of different vitamins. So as your bricks rises, that percent solid, that's what you're actually measuring is that material. And so you are getting higher nutrition, both in the form of nitrogen, phosphorus, sulfur, magnesium, calcium, as well as the sugars. So you want to understand that both of those things are components of bricks when you're measuring bricks. Okay?
All right, fantastic questions. Looking over some of the questions that Neil sent to me from a lot of you, a lot of people are asking about the compost injection, compost tea injection equipment. And Neil, Elaine, you can find that on Environmental Celebration Institute, right?
Right. We've been working with Grow King, a company that really wants to have the equipment for doing all of the mechanical applications of biology. And so they are developing very small size units for homeowner gardens, people who have just 10-foot by 10-foot areas, or an acre or two, small size equipment for that kind of market, where both spraying the foliage above ground as well as you just change the handle and you go from your above ground sprayer gun to your injector, and very heavy duty and very well made. So I really like what Grow King has been doing. And the place to get the information about that is on the environmentcelebration.com website. Or if you can't find it, because we just shifted over and it's all new website, in case you want to go look at it again, and leave comments on what you think needs to be improved. But I think pretty sure that Grow King is sprayer is on the website. So you can go take a look at it. But the person to contact about the Grow King sprayer is Michelle at environmentcelebration.com, and she can send you all their sales information. So you can see, what's the pressure and what's the size of the pump and what's the battery and how do you move your tank around. It's got this cute little cart. And now they're going to larger size units. 100-gallon, 250-gallon, 500-gallon spray units. So those are the people to get hold of to find more information about this.
All right, awesome. So yeah, if everyone's looking for the link, it's down in the chat box below, environmentcelebration.com. It's also the link to those classes with the discount available till tonight. Taking advantage of that. So now we're on to the compost tea questions. Rob asked, it's funny because there's 6 or 7 Robs in the chat on the webinar right now. Rob asks, I own a lawn fertilization company and we have been applying compost tea along with some low salt fertilizers for a year. Is it possible to get the soil to a place where we could only use compost tea and no fertilizer? If so, has Elaine seen this before and how long does it take?
Definitely you can get to the point where you're not using any inorganic fertilizers, and usually if you're paying attention to the biology and working to move it along in the right balances for the plants you're trying to grow, it's one growing season, maybe even half a growing season you'll get there. But definitely by the end of one growing season, my expectation it would be that you have the biology in order. Now, admittedly, we work with some growers who have preconceived notions about compost and how to make it and what should work better, and they don't listen to us. And at the end of making a batch of compost, and I look at the data from that compost and I go, what did you do? And then it starts coming out, well, they didn't think that this was important, so they ignored the whole business about 50% moisture. Or they didn't think that I really meant that they had to take temperatures every morning on their compost pile. And of course, I do mean that. So how else can you figure out whether this thing is freezing to death or it's got a fever and everything's about to go kaboom? So we've got to be monitoring things. So as long as you're paying attention and you're moving the biology in the right direction, then it's going to be successful within that one growing season. So if you have growers who want to go a little bit slower, it's their choice. They could continue putting on some amount of inorganic fertilizer. They could use a pesticide or a herbicide spray if they're seeing some problem because they don't have the biology converted over yet from dirt back into soil. They could well see some weed problems and some insect problems. So, okay, they're not quite there yet. So it's really when when you have, enough, I'm gonna get the biology right. Once you get the biology right, then no, why would you be out there putting on more? Well, some people have to be doing something. So if you feel the requirement to continue putting out more and more and more and more good biology, it's not going to hurt anything. You're not going to have a problem because you keep enhancing that good biology. But when you have more than enough, most of that biology is just going to go sleep and wait until they're needed. So hopefully that answers that question.
Yeah, hopefully let us know what you thought. All right, so next question is from Mary. I think it's Mary Combs. This is she says, my question is quite specific. Northern Idaho suffers from Amarilla root rot and it is a problem on my property. Is it possible to outcompete Amarilla and other root rots with good guy fungi? Can I improve the chances of our trees fending off root rot through application of compost and compost teas?
Yeah, and we may suggest some specific microorganisms to put into your tea to specifically go after any particular disease-causing organism. So, Raleigh, I don't see where we are on these questions. So I'm wandering through.
Oh, I apologize. This is one I added in. Like Neil sent me 20 questions like in the morning, so I just added in a few that people sent. So apologies.
Because I'm wandering around going, where is this question?
What's going
A
on?
bunch of people wrote me and I sent it to Raleigh this morning.
Yeah.
Okay. So you'll have to tell me if I answer all of the questions. I will do my best to remember all the parts. You may have to clue me in. So yeah, is it Amarita? That is the particular fungus that she's talking about?
Yeah, it says Amarilla. It's like an armadillo, 2 L's. Armilla.
Armillaria.
Yeah, Armillaria.
Armillaria? Okay. Yeah. Okay, that's a bad nasty. And so with that particular guy, what you want to be doing is injecting down into the soil, because what you have to do is put competitors of that fungus into the soil. So in the root system itself, you can get this war starting to be fought. And then, we'll stop seeing the fruiting bodies above ground. You'll know that the plant is healing, it's recovering from that problem. You then want to make sure that you maintain that good biology around the root system, and so that the disease doesn't come back, because that's a big problem. You've got the spores, you've got the hyphae, there's constant warfare is going to be happening in that root zone for probably 15, 20 years. And you just got to stay up on it. Because any kind of root contact of an infected tree could transfer the infection to all the surrounding trees. Number of different fungal diseases that behave that way. And so any one of those, you want no root contact of the infected root system. So you got to treat the whole root system. So did I answer everything for that particular question?
Yeah, I think
you did.
Okay.
Is
Awesome.
when you say that's a bad nasty, is that the scientific term?
Oh yeah, highly scientific.
That falls in the category of bad
It's
nasty.
the worst. The
Yeah.
worst.
That's what would the proper scientific term be? It's an aggressive fungus.
There you go.
Ooh,
Okay.
don't like that. Okay, so I think that's through the compost tea questions. We're gonna have to kind of get through these here because we're trying to squeeze out the last remaining, 26 minutes. There are some questions about compost tea equipment, but I'm sure you can find answers to a lot of those on the Environmental Celebration Institute site. So we're moving on to specific questions about the Soil Food Web science. These are questions about the different chemical compounds, the interactions in the soil. So the first one, this is from Maureen. Let's see if she's here. Maureen asks, I've heard it helps to add clay to decomposing granite to improvement. She lives in Southern California, and she's talking about adding to a relatively small home. What are your comments?
I'd like to see their data about why clay would help decomposing granite soil. What exactly would be improved by adding clay, other than you're adding very small clay colloids? The granite is probably big chunky things, and so adding the smaller particle sizes might allow bacteria to start building soil structure. Bacteria do need small things to build those micro aggregates. But what makes more sense to me is to add organic matter. There's an equal surface area with clay and organic matter. And at least with organic matter, most microorganisms have an easier time pulling the required nutrients out of the organic matter. I always kind of, get all, when somebody says clay has all the nutrients your plant requires. Well, yeah, that's true, but it's in crystalline form. And therefore, you're going to have those microorganisms being fed exudates from your plant or other organic matter in order for them to make the enzymes to pull the crystalline material apart and pull those nutrients into the body of the bacteria and fungi. It's a lot of work. So why not just put compost on, or mulch where you've mixed in a little bit of compost where you've got the organisms, and then build that decomposed granite with that organic matter and build a really good soil in a very short period of time. If you just add clay, you're not building soil. You're not putting any food sources into that soil to help build that soil and end up with some pretty good soil for growing whatever you want to grow. So with the semi-arid climate in California, you have to put the organic matter back into that soil in order to build the structure so you can hold water in that soil. When you go back to the pioneer journals, where the first pioneers, the first people starting to settle in California, their descriptions of the soil in the coastal regions of California were just, this is the best stuff. This is black gold. This is just magical. We just have to put our seeds into the soil and we get the largest plants we've ever seen, bumper crops every year, year in, year out, for somewhere around 15 to 25 years. after they started farming that soil, but they weren't replenishing the organic matter. And so they slowly and surely did what all agricultural practices do, is mine out the organic matter, blew it off as carbon dioxide, took nutrients out in the crops that they grew with absolutely no replenishment of the biology. And so as soon as they start running out of fertility, they start putting manures on. Manure is very high in salt. They started grabbing kelp from the coastline, and they didn't wash the kelp well enough, so they were putting salt on. So of course it turned it into a horrible, well, it's not really even dirt, it's decomposed granite. All of the organic matter is gone, all the clay is washed away. How do you fix this and bring it back to a highly productive condition as rapidly as you possibly can? Compost. Get the organisms in there, rebuild that soil structure. I drive USDA extension agent people crazy, because we'll start out with the soil that's absolute sand. I will get the USDA extension service person to come out and say, yes, this is just sand. Okay, great. Come back at the end of this growing season, please. We put in real good compost, a ton per acre. You may put a little additional organic matter back into that sand, till it in, start growing our crops, 2-3 applications of compost tea. On the field, we invite the USDA extension service person to come back and then tell me what kind of soil this is. And they go, oh, that's a gorgeous loam. Look at the structure in here. This is fantastic. No wonder you got such great crop production. You know, you surpassed the county average. You're up there. You're the best grower in this county when they sit down and actually look at the data. And so, you know, I'm like, okay, so see what we can do with biology. We've taken this sand and turned it into a beautiful loamy soil. And so the extension service guy asks me, so, okay, where did you put the sand that you took out of this field? Where did you dump it? And where did you find this really good loam soil to put back into this field? And I usually just look at him like, what? I don't have the money to do that kind of thing. No, we took that sand, added the organic matter, and we have made that sand behave like a loam with nothing more than the addition of good compost. So we could do that everywhere. It's not magic. It's just that you need to understand your biology. Okay, enough for that question.
All right, great answer. Okay, we've got 2 remaining soil science questions from Astrid and Brianna. They've been very patient, waiting around. So why don't we get to the really heavy one? This is a pretty in-depth one by Astrid. Then we can quickly touch on Brianna's and then get to the few remaining site-specific questions. So Astrid asks, does the plant you plant signal what microbes grow in the soil rhizosphere, or do microbes you inoculate the soil with determine what plants will grow well or not? Okay, this is a very expansive question, Astrid, and the second part is, oh boy, should I just say it? Does the plant you plant signal what microbes grow in the soil rhizosphere, or do the microbes you inoculate the soil with determine what plants you grow well or not? For example, when building soil, raised beds with compost straw and not tilling, a high fungi to bacteria ratio has been created. Is this a case for tillage and inoculating with a high compost tea to mitigate against the tilling, or will the plants support the growth of the microbes they need by the way of their exudates? And then she go into how the work of Carrie Ream supports this.
Okay,
big question.
Yeah, this one also, this usually takes some time. So that first part, does the plant you plant signal what microbes grow in the rhizosphere, or do microbes you inoculate determine what plants will grow? And the answer to that is yes, because it's both. And it's always it's important to understand how much role the exudates coming from the root system have on what microorganisms will grow and prosper in that root system. But it's also important to know that the balance of the fungi to the bacteria very strongly influences what plants can grow. Well, okay, so how does it work that way? When you initially plant that plant, you want to make certain that the forms of nitrogen in that soil are going to promote the growth of that plant. So most people think of just nitrogen. We've got nitrogen in our soil. No, you don't just have nitrogen in the soil. That nitrogen comes in a particular form. So is it present mostly as nitrate, NO3, or is that nitrogen present mostly as NH4, ammonium? And it's really important to look at that balance, because if you've got mostly nitrate in your soil, it means, where does that nitrate come from? It comes from the actions of bacteria. Aerobic bacteria produce a lot of glues because they're making all those microaggregates and they're gluing the microaggregates together. And the pH of those glues produced by the bacteria always have a pH greater than 7. So your soil's alkaline. I always love it when somebody says, no, Elaine, you don't understand. When plants are taking up nitrate, it's causing the soil to become acidic. And I look at that one and sort of like, what? If that was true, then all of the soils in the Great Plains of the United States should be acid, because we've got massive root systems of grass, trees, and shrubs in the Great Plains of the United States, depending on exactly where you are. But think about a grassland, massive root systems in there. Every bit of that soil is occupied by roots practically. And yet, what's the pH of those soils in the Great Plains of the United States? Well, the pH is going to be up around 7.8, 8.5, 9.0, somewhere in that range. Yes, and that's alkaline, isn't it? If, when your plant took up any form of nitrogen, it released hydrogen, it acidifies your soil, then how come the soils in the Great Plains of the United States still be alkaline? Well, it's because you've got calcium in that soil. You have calcium everywhere in soil. There is no soil that doesn't have calcium in it. So how can it be working some way, this way right here, but very different ways over there? What? You've got to understand the microbiology in that soil. Bacterial dominated soils, mostly those glues are going to be alkaline, and therefore that's what's causing the pH of that aerobic soil to have an alkaline pH. That means nitrifying bacteria are going to be growing and expressing their enzymes that will immediately take any NH4 that's present, that's released when bacteria and fungi are being eaten. What the protozoa, nematodes, microarthropods, earthworms release is NH4. If you're in an alkaline soil with the nitrifying bacteria producing the enzymes to rip the hydrogens off that NH4 and convert it to NO2 and then NO3, then the predominant form of nitrogen in a bacterial dominated soil is going to be nitrate. You set the stage to grow weeds. So if all you've got are bacteria, you're going to be growing a boatload of weeds. This is going to be a horrible soil. You can't possibly retain the nutrients that you want. Well, okay, you see where we end up with weeds. Now, if we can get a little fungi growing in that soil, now the fungi will produce that acid condition. The fungi don't produce copious amounts of glue. They produce organic acids, but the pH won't drop below a pH of 5.5. Only if you go anaerobic. Anaerobic bacteria, billions of them growing, maintaining things anaerobic, yes, you will drop your pH down to, I've dealt with, I would call it dirt at this point. I've dealt with dirt that has a pH of 0.5. You are not growing anything in that soil, weeds included. So think of the gamut that we can go through when we're trying to understand pH. So fungi become starting to increase that fungal component. They're going to be developing NH4 and ammonium pool, which is going to select against weeds. Weeds don't utilize NH4, and so other plants are going to win in competition because now they can get hold of that NH4 and start outcompeting the weeds. So as we move through succession, the amount of fungal biomass increases and increases and increases because there's more and more and more woody component in the plants that are growing. So when your plant first goes plunk into that soil, if you put a start into that soil, you put a seed into that soil, and the seed starts to germinate, it's immediately impacted by that bacterial fungal ratio, by the nitrate ammonium balance, and that immediately starts controlling whether that plant is going to be happy or whether it's going to be kind of stressed or very stressed or completely stressed. At the point that the plant starts pumping exudates out of its root system, so that means it's got to be in a place where it's comfortable, where it's not being stressed so badly that it can't even grow or it's not doing well, everything else is out competing it. So as long as that plant is healthy, which means it's already having to deal with that fungal to bacterial biomass ratio. Now as nutrient cycling starts, and now we got protozoa, nematodes, microarthropods, earthworms, all of those things cycling the nutrients, the plant is putting out the exudates to tell which bacteria and which fungi it wants to grow. Because those bacteria, those fungal species make the enzymes to pull the nutrient that the plant needs out of the sand, silt, clay, rocks, pebbles, and organic matter. So both things are an aspect of which plants are going to be happy, healthy, and able to outcompete everything else. So it's not just one or the other, and that's what you'll notice in nature is things don't operate on just one way. There's a number of different things contributing to the general health of the plant that you want to grow. It's not just one factor that you've got to pay attention to. You have to pay attention to everything. So if minerals aren't balanced, you can't balance minerals by putting in soluble inorganic nutrients. You can't do it. You don't know what the plant is lacking today, right now. So if you're putting in inorganic fertilizers right now, you're messing up the metabolism of your plant. You're screwing it up because you got it wrong. Now, well, won't these soluble nutrients only be taken into a plant at the rate the plant requires? No, because you've messed up the concentrations of the chemical form, calcium nitrate or potassium nitrate. If you put in such a massive amount of nitrate into the soil, you're forcing that plant to take up the calcium or forcing it to take up the ammonia, the potassium or the calcium or whatever else that might be bound to. You're messing up the metabolism of your plant. So we don't want to be pushing soluble inorganic nutrients. We've got to let the plant determine those balances. The plant knows what it needs. It will tell the bacteria and fungi what it needs. And then that attracts the protozoa, nematodes, microarthropods. And so the plant is getting exactly what it needs, all of the nutrients in proper balance. Not just one nutrient or two nutrients, it's all of them. How many nutrients does your plant require? All of them in the right balances. Do you know what your plant is lacking right now? And guess what? Right now it's lacking something else. And if it's in a healthy soil, it now needs something else. And it's that fast to turn around. If you take a soil sample, if you take a plant tissue test and you go get it analyzed, you're going to get the data back when? 2 weeks from now, 3 weeks from now? Is that telling you anything about what your plant needs 3 weeks from now? So let the plant take control of its own life. We have to set the stage so that plant will be healthy. But it means we set the stage and then let the plant direct the action on that stage. We have to stop being so arrogant that we think we know better than the plant what it needs. Okay, end of that.
Ooh, boy, that was a heavy one.
I'm gonna say goodbye because I've got a vet appointment that I've got to take my dog to. But I just want to plug for this Thursday. We have another microbiologist coming on. That's Walter Jane. He's an Aussie fellow who's going to be talking to us about the small water cycle and hydrology. Going to be really, really good. And then if we didn't get to your question, because I think we've got more people writing in on this sidebar every couple minutes. We send it to us by mail, and we'll do our best to get to you. But that's it for me. So thanks, Elaine, for coming on again. Thanks to the 120 of you that showed up, and we'll see you on some future webinars. All right. Thanks, Raleigh. Also want to shout out Raleigh for sending all us an awesome job.
Oh, thank you.
Thank you much.
Take care, all.
Hope your dog is good. Okay, so I know this is tough. Well, Piang said his mic had a harsh metallic tone. Well, hopefully next time his mic will be smoother. All right, so there's a few more questions. This is tough. I mean, Elaine, we go only as long as you want to. We set an end at 1. However, if you want to stay a few minutes longer, it's only dependent on how long you want to stay. But how well, there's another good soil science question, but there's also a lot of site-specific questions. So I at least want to get to one of those. So let's move to a site-specific question. Here's one from Betsy Flack. So Betsy says, we have 10 acres of hay fields and a conservation easement that prevents tilling or deep disruption of the soil. She says she has a hard pan about 12 inches down, and there's a lot of weed grasses such as velvet grass, Brizia major. She says, how do we deal with the thatch so we can drill seed and also apply the principles of composting to create the meadow without tilling and ultimately reintroduce native grasses? And she's in her 70s, so she doesn't want to do too much crazy hard work.
She doesn't want to go dig up the whole field. So no, I have the same attitude myself. How can we put these microorganisms to work for us as rapidly as possible with as little work as possible? And so the description of what she's giving is she's got, and I'm kind of asking you to correct me if I'm wrong here, Raleigh. She's got a mulch layer. She's got a thatch on that pasture or meadow. That's how thick? Did she say how thick?
She said she has a hard pan 12 inches down, and on top of that hard pan layer on her hayfields, she says she has a lot of weedy grasses such as velvet grass and Brizia major, and she says those are taking over. And her question is, how can we deal with the thatch so we can drill seed and use composting or to reintroduce native grasses?
Okay. What I missed was how thick the thatch layer was, and you were breaking up when you were rereading that question. So
okay, sure. I didn't see anything about the thickness of the thatch layer. She just says, how can we deal with the thatch so that we could drill seed and apply these principles of composting?
Yep. Okay. So if this was last fall, I would say the simplest thing to do is to either go out and spread a ton per acre of good compost on top of that thatch, and the biology would work its way through and have that thatch completely decomposed by the beginning of this next growing season. So during the middle of the winter, and her ground is probably frozen at this point and possibly even under some feet of snow, then she's going to have to wait until that snow starts to melt in the springtime. And I would probably go out with a spray of compost extract. She doesn't need to make a tea, just an extract. And apply it just as the snow is getting that rotten aspect where it's all kind of mushy and breaking down, corn, snow, whatever you want to call it. It's rotten. It's about to melt, because that's a really good time to get this biology back into the soil and get it into that surface, that thatch layer. So immediately that thatch is breaking down. Then come the time when the plants are starting to grow, they're flushing, getting the stored nutrients from their root systems and starting to grow the above ground part of the plant. I would go out and put on a second application of a compost tea. So this time a tea, because you want the organisms to stick to the leaf surfaces of the plants and protect the above ground part of that plant. You're going to get biology onto the surface of the soil and onto any of the thatch that did not decompose. And then that starts building the structure in the soil. You might want to go out a month later and a month after that. You don't want to be applying biology to the soil when it's getting really hot and dry. It's kind of like, think about yourself. If we were going to miniaturize you, and was that movie Downsizing or whatever? If we were going to make people really miniature and then kick them out the door of the airplane as you fly over the grasslands, would you survive once we get into typical July, August, September weather? It's bone dry. The soil surface is hard as rock, but if you don't have the proper biology in it, you may have a thatch layer that's really nasty, probably not going to survive. But if we drop you off in the springtime when there's lots of water, everything's nice and green, lots to eat, no problem at all. Same thing for your microorganisms. Just think of these creatures in the compost, in your soil, in your teas and extracts as just being like miniature human beings. If you wouldn't do it to your children, don't do it to your microorganisms. So get those out on the soil. It's probably going to take at least 1 to 2 growing seasons for all of the benefits to be established in your soil. You might want in the fall to be checking the biology in the soil, make sure that it's being improved. Get that metal penetrometer out and find out if you've broken up any of your compaction layers. So if you can this spring, go out and find your compaction layers. When your soil is very wet, you're only going to find the really, really nasty, the really, really bad compaction layers. As your soil starts to dry down into that dry summer period, you're going to be picking up all the rest of them. The rest of the compaction layers that are just due to bridging factors that start to occur as you have salts that are being accumulated in that soil. And so you'll find a whole bunch of extra compaction layers as you go through that growing season. But so for pasture systems, that's probably all you need to do. There are a number of places where I've worked in Idaho, Montana, North and South Dakota, Iowa, Minnesota, New Mexico, Arizona, Colorado, where when we put the biology out on the surface of the soil, an application of compost extract or tea, we have compaction layers. When we started, there were compaction layers at 4 inches, at 8 inches, at 12 inches, at 16 inches, at 30 inches, at 4.5 feet down. There's just every few inches there was another compaction layer. One application of compost extract to the surface of the soil, and that all got dealt with. So the next time we came out, approximately 6 weeks later, there were no measurable compaction layers in that soil. The soil was drier than we had tested initially. So I was expecting way more compaction layers, but all the compaction layers were gone. They had been broken up by the biology that we had added that one time. So if you're really, really lucky and your soil is really not that bad off, hasn't been treated that bad, it can be that easy to fix the problems. On the other hand, maybe unbeknownst to you, they were out there doing sheep dips in your pasture, and they were doing all kinds of horrific things out there in your land, and they never told you. You have no idea, and it's going to take maybe 2 years to get things back on track and functioning correctly. Okay?
All right. Yeah, great question. All right. So it's up to you, Elaine. We could either end on one final question or two, just depending on what you want.
Let's go with two.
All right, these two. Okay, so great. These are more a bit more simple. These are just more general advice. The first one's from Daniel. It's about pigs. So Daniel's asking, I know that tilling is bad for the soil food web, but I have a piece of former hayfield that I want to plant small grains on. Would opening up the ground using pigs be any less damaging than machine tillage?
Actually, pigs are a lot less damaging than mechanical. They don't do the pulverizing characteristics that our discs or chisel plows or even subsoilers do. So pigs are a lot less problematical. Are you still there?
Oh yeah. I'm
Okay.
here.
My computer did something strange, and I'm going, oh no, I lost you all. No, you're all still there. So I would probably prefer that you put pigs out if you possibly can, because at the same time that the pigs are out there rooting around, they're pooping. And that manure is a pretty good addition, because they're going to happily mix all that manure in, and all of the biology. Yeah, it's not the greatest biology in the world, but if you have also then added compost before you put the pigs in there, go out and spread your layer of compost out on that property in that area you're going to put the pigs. Let the pigs go in. I generally will, throw out a handful of seed corn or some kind of very small seed that the pigs absolutely love, which gets them to root around and do an even more complete job of breaking open those top 5 to 10 inches of the soil at the surface. Especially if you've got thatch at the surface, the pigs are going to do a great job of incorporating that organic matter into your soil.
All right, awesome. Yeah, using pigs is great. I remember at Versaland Farm with Grant Schultz, they had this big overwintered pig area, and as soon as they moved the pigs off in a few seasons, like there was just an explosion, like the grass went up, and then they're rotating the pigs, and it was just great seeing how all those areas evolved compared to the monoculture corn crops next door.
Yeah. So pigs are very nice because they leave behind urine and poop.
Oh yeah. Okay. So next question. I think this is the final question. Final questions from Julia Britton. Let's see if she's here. I'm sorry, Beverly. Brianna, I couldn't get to your question. It's really good, but alas, we just don't have time. It was about, she wanted you to respond to your defenses of the soil minerals question, but
yeah,
Julia's...
I usually just go and get a couple more tables, like the one I showed. Any introductory soils textbook has basically that same chart showing massive concentrations of every nutrient in soil. Everywhere on the planet, there is no place that lacks nutrients. So how could anybody defend a different point of view? You have to be careful because soils ignore the total amount of nutrient. And that's what I'm talking about, total nutrient content. They've been for the whole entire life just on the soluble. That's all they know about. Well, soluble nutrients are... Okay?
All right. Well, you caught out a little bit at the end there, but I think the point was had. That was a great a great little catch up on there. So the final question is from Julia. She's saying, could you please advise us on whether tarping the soil to prepare a seed bed is a good idea to help get rid of crop residues and cover crops? Because this is the advice of a lot of market gardeners that they advocate. And her other question is, would it be better to use landscape fabric instead to allow oxygen in to keep the soil aerobic?
Yeah, it depends on what kind of climate you're in. Quite often you get advice on covering the bed because you're in an arid climate and you've got to do everything you can to keep the moisture in the soil, and you don't want all that evaporation. Well, if you can get some cover plants and grow cover plants, that pretty much drops that evaporation down to just about nothing. People will then always say, well, yeah, but plants do transpiration, and so you're going to lose more water due to transpiration. Well, not true. Only if you're growing weeds. Yes, weeds are horrible at holding on to water. Weeds, what do they care about how much moisture is still in the soil? The weeds are going to grow very rapidly, reproduce very rapidly, and so what do they care what's happened to the water? If you're putting in low-growing cover plants, they're actually reducing the amount of evaporation, and there's been a bunch of studies done up at Washington State University showing that fact over and over. Yeah, if what your test plants are in your system are weeds, yeah, you're losing more water by having those plants on there. But any later successional stage, you hold more and more and more water. Keep it, prevent it from being evaporated. The later in succession those understory plants are. So, and your bed with that. Now, what are you going to do with the black plastic? It is not sustainable. So instead of covering with black plastic, cover with some cover crops, with some, not cover crops. They have gone the wrong direction with cover crops because they have selected for taller and taller and taller plants where more and more and more biomass is being grown above ground. Where do you need this biomass? Not above ground. If you cut it when you've got 12 feet tall Sudan grass or something, now you're going to cut it and it lies down onto the surface of the soil. How thick is that layer? That's going to be a huge problem. It's probably going to go anaerobic. And now you're causing all kinds of damage to all the biology and building structure and having the right pHs and all of that. It's just the wrong direction to go. So instead, let's grow really short, low-growing plants where they have root systems that go down, 10, 15, 20, 25 feet. And so you're putting all that good food into the soil, maintaining infiltration of your water so that all of that depth of the soil, water is being stored for your root systems of your crop plant to go down and get. So let's establish those understory plants so we can get water storage, so we can hold carbon in that soil, so you can maintain air infiltration, root systems go deep, so you don't have to worry about irrigating in the summertime, and then you don't have to play the black plastic game to try to hold moisture in that soil. You won't have thatch developing because you're going to maintain that really good biomass of microorganisms that will very rapidly decompose anything that falls to the surface of the soil. If you're seeing plant material lying on the surface of your soil for more than a 4-week period of time, get the message from Mother Nature. There's something wrong with the biology in your soil and you need to fix that. So learn to read the information that Mother Nature is putting in front of us. She doesn't speak to us using English. She speaks to us by putting information right in front of our faces. We just need to be able to interpret it correctly.
All right, Elaine, thank you for sticking around for 2 hours to answer so many awesome questions. We had over 80 people submit questions. It's tough. There's so many good ones. And to narrow it down to the 50 that we can answer is tough. And if you have any more questions, we will do this again in the future. This will be exciting. And I appreciate everybody's time. We had almost, I think, 150 or more. I think, how much we had? 1.200, but lots of people showed up again to learn about soil. And if you want to learn further, Elaine has her classes, the Life and Soil classes that offer. The half-off offer ends tonight. So check your inbox, because we will send the replay link to you. And again, appreciate sticking around from learning with Elaine, because we all got to learn about soil. It's how we can survive into the future and make our lives better. So thank you, Elaine, and thank you, everyone, for joining us today.
All right, everybody. Thank you so much for being with us. Hope you enjoyed this, and also check Elaine's courses. Check out her website, see if you like it, and let us know.